Awesome. Thank you, Mandy. So again, I'm the Regional Wildlife Manager in Region 6, and I get the opportunity to talk to you all a little bit about Fisher this evening, particular about one project that I was able to work on between 2018 and 2021. Just as a heads up, I don't see the slides yet. Oh, there we go. Perfect. So before we get into that, I think generally most folks probably in on this call have an understanding of what a fisher is, but maybe not. So we want to clear that up before we dive in any further. But identifying characteristics, fisher are a land-bearing species. They're dark-haired member of the weasel family, large as compared to a weasel or a mink. As you see there below, males are between 7 and 15 pounds and females weighing between 3 and 7 pounds. So some of our females do get confused with other mustelid species like a mink, like a large adult male mink could be close to the size of a female fisher. And then same with marten in that range in the central Adirondacks where marten and fisher overlap. If you have a small female fisher, there could be some confusion with a marten. But their color does range. It can be very, very dark black to a light grizzly brown. A lot of the fisher, they have that kind of patch as you see in this photo where there's some white. I've also seen it where it is even kind of orangey colored, but generally white because that orangey color is kind of distinctive of marten as well. But we can move on to the next slide. So common misconceptions. There's a lot of them wrapped around fisher. I feel like especially in Western New York where fisher over the last decade or more have been expanding their range and they're the new kid on the block. So they kind of get blamed for everything wrong going on, right? It's kind of how it goes. But overall, their diet, though they're known to be vicious killers or that's what people say, they're opportunistic omnivores. They have a diverse diet, includes small mammals, birds, fruit, nuts, and more. Doing the research that we did, we've seen fisher crawl up and pluck out young in the year wood ducks, have them walking around with apples in their mouths, walking around with chipmunks. I've seen a den that had a deer leg in it. So I want to be clear, they're not taking deer, but they're scavenging on them as well. So they're opportunistic and they're going to get their mouth on anything that they can. Fisher cat, a lot of folks call them fisher cats. They're nowhere near and related to a cat other than they don't like water. We have seen videos of them swimming across bodies of water, but if they can avoid it, they will do so. So maybe that's why they got that designation as a fisher cat, but nowhere near closely related to a cat. And then lastly, screaming fishers. We've all heard that. You hear that noise out there, that's one of those crazy fisher. So, but not necessarily. Fisher do have some vocalizations. Maybe that's another reason why they kind of were associated with a cat, because it's almost like a purr when you have them in hand, which most people are not going to have them in hand. But they do not scream or yell as most folks might assume or have heard in the past. So that is in all just kind of generally what a fisher is. So next, I'll just do a brief summary on some work that we did in the northern zone of New York. We started this project because we collect, as some of the data that Mandy had talked about that we collect from trappers, we had some data that suggests maybe our populations in the Adirondacks were declining. And as a result of that, we did reduce season length in the Adirondacks for trapper fishing or fisher trapping, excuse me. But we wanted to learn more. We wanted to know, was that change justified? What's going on with the fisher in the central Adirondacks? And to do that, we compared the fisher population in the central Adirondacks to the fisher population on the Tug Hill. Reason being, fisher population on the Tug Hill, the data that we had at the time showed that it was a robust population, though some of the characteristics on the landscape between the Tug Hill and the Adirondacks are somewhat similar, right? Large tracts of forest land, similar kind of latitude in northern New York. And we want to kind of see what was going on between those two populations. So as I explained, the study was a comparison between the Adirondacks and uh, the Tug Hill being on our left-hand side of the screen and the Central Adirondacks being on the right-hand side of the screen. The blue dots that you see are the number of animals that we captured. So to do this study, we had to rely on contract trappers or general fur trappers that we utilized to capture animals. We capture them in tomahawk live traps. Trappers would let us know that we got a fisher. We'd run out, grab them, immobilize them, put a collar on them, take some other biological measurements, and then release them back in the same location that they were trapped. And by doing that, we were able to get the information that I'll be able to explain here in another couple slides. So overall, between the two study areas, between our efforts and also contract trapper efforts, we trapped close to 200 animals, 30 females, 50, well, between the two areas in the Central Adirondacks, there's 30 females, in the Tug Hill, 51 females. And then you see in the Central Adirondacks, 48 males, and 48 males on the Tug Hill. And if you look, it's kind of interesting, and I'll get into a little bit more, but just looking at this data right here, females drive populations. You need females to continue that population. And just looking between the Central Adirondacks and the Tug Hill study area, just our sample that we collected, the Tug Hill had more females, which is kind of interesting. Something that we didn't necessarily totally dive into, but a fact out there that's kind of interesting that there's more females that we caught on the Tug Hill. All right, so the first thing that we were looking at is overall adult survival. When we capture these animals where we were pulling a tooth, that would help us identify how old they were, so we could determine survival rates between male and females, adults and juveniles. We could try to identify what could be limiting their survival across the landscape. Like, are the fish in the Central Adirondacks, are they overall succumbing more or as adults versus the Tug Hill? What's going on? And then in every wildlife research, the gold standard or the hope is like to identify specifically what they are dying of. But that's always easier said than done. But we did get the opportunity to identify, in some cases, what was actually killing the fish on the landscape. So with that, the Tug Hill survival rates of the 99 individuals that we monitored on the Tug Hill, 9 were still alive in December of 2022. So that's when we ended the study. 34 went missing or their fate was undetermined. Some of these animals, we were fortunate enough to track some juvenile females. It seemed like juvenile females would travel a lot more, but they traveled the entire Tug Hill. We had some in the Adirondacks that traveled through the entire Adirondacks. And sometimes, though we use flight and also other means to track these animals, sometimes they get lost. But another factor is a lot of times these radio collars, you put them out and sometimes they don't work either. So unfortunately, 34 of them, we really don't have any ideas. We could just assume. 39 died. 7 were released without a collar, at least in the Tug Hill area. That could be there's an injury or something happened that we didn't feel like we should have put a collar on that animal. And then 10 slipped collars after release. And that's very common for females. For those of you who handle mustelids or know anything about mustelids, they have a real streamlined body. So the collar has to be just on there perfect, where it's tight enough not to cause any injury, but also tight enough where it's not going to fall off. So it's a really fine line. And sometimes with smaller females, those collars would fall off. So causes of death on the Tug Hill, which this is a really interesting piece of data that you should remember for the next slide, is 24 were from regulated trapping, 4 from rodenticide poisoning. And that is folks going to, various hardware store and purchasing those little bait drops and put them, in your yard or in your camp. Mice are eating those. They're going out dying. Fish are eating those mice, and they're bioaccumulating that rodenticide enough where it can kill them or cause injury. So don't do that if you can't. One from rabies, which is the picture here. I was able to climb up this tree and find this fisher, which is amazing to find a fisher that, isn't just bones. And it did end up testing positive for rabies. And then 10 were undetermined, which are pretty much those bones that we come across. In the Adirondacks, of their 78, 14 were still alive in December 2022, 27 went missing or their fate was undetermined, 25 died, and 12 had slipped collars before December 2022. Their cause of a death, so note 4 from regulated trapping, and that was a comparison of 24 in the Tug Hill, 3 from vehicle strikes, 2 from predation, 1 from ammonia, and 15 were undetermined. The picture there, the thought was that was preyed on by a coyote. Moving on, so that was what we talked about survival. So one part of that study was adult survival, seeing what's happening with those adults. The next, and really the most important part of this study, was trying to figure out reproduction and how is that going to change between the Tug Hill and the Central Adirondacks. And then how are these different factors in forest composition or prey base affecting that? Or could we make assumptions based off of what we're finding from the study? And with all these adult females, so when we have them in hand, we're identifying how old they are based off of their teeth structure and tooth wear. So we have a pretty good idea what's going to be an adult female, and those adult females got a very specific GPS collar where we could track their individual points, but it also had a component where we could track them to find their den trees. And I'll explain that in the next couple slides on why that is important. So why that is important is because this is how we collected that reproductive data. So how many kits a female was having during the spring. That is the reproductive data that we are trying to achieve. So how did we do that? So we collared these adult females, and we are doing our trapping in the winter. Come springtime, which fisher in northern New York, generally they're having young around April 1st. Once we could monitor their GPS points, but it was best for us to go out early in the morning. And as we went out early in the morning, and there was that fisher was up in a tree, in the same tree a couple mornings in a row, we assumed that she had a litter there. Once we identified that she had a litter in that tree, or we assumed that she did, we would set trail cameras around the base of that tree. And then confirm that she had a litter, because fisher will move their den trees 4 to 5 times throughout that rearing period. So from April to June. And when they're moving those trees, because they're, they may start in a really small, tight hole. And then as the kits get bigger, they got to move on. And they're also moving on to stay ahead of other predators or other male fisher. So when they move and go down the tree, they're moving one young at a time, and we were able to capture a photograph at the base of the tree when she was moving. So that's that photo in the middle of the screen, also the photo on the bottom right. And then we just monitor them and kept doing this until about the beginning of June, when the kits were able to move on their own power. And once they're able to move on their own power, they're less likely to use one specific den tree very regularly. And we lose our ability to track them as we did early on. So with that, the Central Adirondack study area, they were able to monitor 10 females on the Tug Hill. We were able to monitor 11. And just note, it took, close to, it's about, for every one fisher that we caught, or every 10 fisher that we caught, one of them was an adult female. So we had to catch a lot of animals to get that adult female that was going to have kits that we're really trying to collect data on. The average number of kits for the Central Adirondacks was 2.1, and the average on the Tug Hill study area was 2.7 which that is a very important piece of information. As you can see, there is a difference between these two study areas. Yeah, perfect. So key findings, wrapping up here, Fisher survival on the Tug Hill is much lower than the Adirondacks, but is offset by higher productivity and kit survival. So we noticed when I talked about adult survival on the Tug Hill, we had trappers take 24 of our animals, and on the Adirondacks, they had 4. So our overall harvest on the Tug Hill is higher, but in the last statistics, I said, the overall kit production for Fisher in the Tug Hill was higher than in the Central Adirondacks. So though we had more mortality in the Tug Hill, that reproductive rate is much higher and is driving that population and making it more sustainable than in the Central Adirondacks. So also across both study areas, Fisher survival and productivity was linked to beach mass cycles. So in springs following good beech nut production, Fisher tended to produce more kits and survival rates increased. And lastly, despite risk of rodenticide exposure, which is putting those little green baits out that causes harm not just to those mice but other species, Fisher survival and productivity are higher in areas with greater wild-urban interface. So those areas that are kind of sort of fragmented, that may produce more prey base, such as squirrels or chipmunks, that seems to be where Fisher thrived, as opposed to a real dense, middle of the Adirondacks continuous forest stand. And that is what I got for Fisher in the northern zone.
Thanks, Tim. I'll take it over from here. So let me just grab presenter mode and pull up the notes here. All right. So we are actually in the middle of a thunderstorm right here in Western New York right now. So hopefully I stay on for my entire presentation. But I am Jen Pettit. I'm a regional wildlife biologist out of the Region 9 Western New York area. And I'm going to talk a little bit about the Southern Zone Fisher camera trap study that we did over the last several years. So a little background on the Fisher population out in central and western New York. Like many species, they were historically found throughout upstate New York, but experienced severe population decline in the late 1800s and early 1900s due to exploitation and habitat loss, and were extirpated from most of New York at that time. As recently as the 1970s, they were only found in the Adirondack Mountains. And in the 1970s, DEC did reintroduce Fishers to the Catskills using licensed trappers to assist with that. A few decades later, Pennsylvania also had a reintroduction program in the 1990s. So the Fishers in central and western New York are coming from 2 different areas, really. So the expansion of the population out in eastern New York, and then also the northern Pennsylvania population expanding northward. So we wanted to get a better handle on what was going on out in central and western New York, and we partnered with the New York Cooperative Fish and Wildlife Research Unit out of Cornell to assist us with that. So in 2013, we launched this study, and we were looking to determine the relative abundance of Fishers, specifically out in central and western New York, with a goal of informing future management decisions. And we accomplished this in a non-invasive technique of utilizing camera traps and also hair snares. So we set up a grid pattern in all participating regions in which we had specific parameters and site locations that we were targeting. And we would sample each site for a period of 3 weeks between the time frame of January through April. So a winter survey. We had you'll see in these two pictures with the fishers, we had a baited tree. The bait was beaver supplied by licensed trappers. So they played a crucial role in our study of providing us that bait. And surrounding the bait, all of the little things kind of sticking off the tree, those are gun brushes that were acting as hair snares. So as a fisher climbs up the tree to eat that beaver meat that is attached to the tree with some chicken wire so that they can't just rip it off, they're also rubbing against those gun brushes and depositing their hair on those gun brushes. So the reason why that was important to us is because it provides even better data. So instead of just seeing that, okay, we have this site and we have a fisher on it, but we don't know for sure if this is one fisher visiting throughout these 3 weeks or if it's many fishers, with the DNA, we can actually look at individual fishers. We also used a skunk-based lure as an attractant. So all of our coworkers and our family really loved, smelling us throughout that study period. And so the first phase, it was a 3-year study from 2013 through 2015. 2013, we were just kind of getting started and ramping up with this. So we surveyed 300 sites in 2013. In 2014 and 15, we surveyed 608 sites. And you might be looking at this map and wondering, why is there that gap at the very southern part of New York near the Pennsylvania border? And that's because we knew from other data that we were looking at, like the observation data that we are collecting from the general public, that area there seemed to have the most robust population. So we were looking to get north of that area and really kind of dial in on, what's going on in that area north and not quite all the way to the Great Lakes Plain at that point. So over these 3-year periods, we tracked where we were getting detections. And you can see on the slide, the red grid cells indicate that we had a positive detection of at least one fisher. The yellow grid cells indicate that fisher were not detected in those locations. And then the gray is just areas that we did not sample at all. And from that, we were able to construct a habitat model that related the probability of the site occupancy to landscape variables. So we looked at a positive correlation to coniferous and mixed forests and some negative correlations to the amount of road density. Also stream density and the proportion of agriculture, that did have a bit of a negative correlation, but not as much as the road density played into. So we were able to construct a habitat model to predict fisher occupancy in the locations that we were sampling, but also across the landscape as well to areas that we had not put cameras at that time. So we also were able to compare areas that were currently open for harvest to areas that were closed to fish or harvest at that time. And after we concluded these initial 3 years of camera monitoring, we put together our fisher management plan, which is a plan for the whole state. So it's not just for Central and Western New York, but in our Central and Western New York component, we proposed opening a fisher trapping season in the area in the orange, which is a season that would have been brand new to us. It is something that we started out in a very conservative way. So it was only a 6-day season. And within that 6-day season, it was very closely monitored. So in order to participate in that season, the fisher trappers needed a permit from us. They also were required to maintain an activity log so that we could gauge how much effort they were putting into trapping. Successful trappers needed to submit a lower jaw because we were able to age and sex that the tooth from that lower jaw, and also have their fisher sealed. So there were several steps in order to go ahead and participate in this season because we really wanted to have a very good handle on what was going on. Also within that fisher management plan, which is located on our website if anyone's interested in reading more about what we plan to do with fisher in New York State, we proposed a second phase of monitoring. So after 3 years of open harvest in Central and Western New York, we went back to our camera surveys and put those cameras back out to see how the population had changed. So we did roughly 600 sites each year. We did modify our sampling design a little bit to reduce the distance between the individual sites in an effort to capture more hair from repeat individuals. So a little bit of a modification, and we did look at some different areas than we had been looking in the past, specifically in areas that had remained closed to harvest. And our key findings from this is that between 2015 and 2021, the fisher population had expanded dramatically, even just in that short period of time, but especially since the 1970s when they were really reduced to just being in the Adirondacks. So they are now considered common throughout upstate New York. They do not currently exist on Long Island. They're also not documented to have been there historically, but in the rest of the state, they are doing wonderfully. And in this figure that you can see here, the yellow indicates the greatest likelihood of fisher presence. The dark purple or black would be the least likelihood. So you can see just from 2015 to 2021 how that's changed. Even though we did open up a trapping season in 2016, the population has still increased.